All right. Hello, everybody. Okay, so it is just about 12:45, so we're going to go ahead and get started. So, yeah, hello, everybody. My name is Trey Britton, and on behalf of our local ASBMB chapter, welcome to this fall 2020 virtual edition of Bears. As many of you know, Bears is normally an in-person event, but we're going to make the best of the current circumstances with this special online version. So there are a few things that we're going to note before we get started. This is one of 3 sessions showing student-submitted research and general science interest videos. Feel free to browse between the sessions if you are interested in seeing videos from the other sessions. And the links are available in the program that was sent out a couple days ago, but let me go ahead and include it in the chat just in case you don't have it on hand. Let's see. Okay, I'm uploading it right now. Okay, so the format is going to be, we have a total of 6 videos in each session, each followed by a brief 2-minute Q&A. Feel free to unmute yourself during the Q&A time if you have any questions for the presenter. And please remember after Bears to fill out the post-attendance form, which I will actually send right now, just in case. All right, so our first video is going to be the detection of protein-protein interaction between the Epstein-Barr viral apoptosis regulator, BHRF1, and Ku70 by Alexandria Mami, under Professor Albert Rivas. All right. So thank you, Alex Umami, for that first presentation. And now we're going to open up the floor to a 2-minute Q&A. So if you have any questions, please feel free to just unmute and say them. All right. Well, if anybody's still thinking of a question, then please feel free to say it. But I will go ahead with a quick question to get it started. So Alex, yeah, y'all had a whole lot of controls that were listed, I think, in that table and in the actual chart. So what were some of the negative controls that y'all used, and what exactly did or were those negative controls looking for?
So basically, the only negative control that we utilized was between RAD21 and Q70, and that was basically to show that if our experimental group, so BHRF1 and Q70, the fluorescence signal that was emitted from that group is not just background fluorescence signal, right? So we wanted to compare that to a negative control to show that there's a significant difference between the relative fluorescence intensity signal between those two groups. And yeah, so the others were just other signals that we just paired with, other groups that we paired those two proteins with. But yeah, RAD21 and Q70 was our specific negative control because these have been documented not to be interactive partners.
Okay, well thank you for, elucidating some of that information. So I've actually one more question. So I apologize if you went into this in the video, but I I'm curious. So what if there is this protein interaction between BHRF1 and Q70, like what's the big picture? Like what exactly does that mean for the project if there is an interaction?
Right. So one important implication really that I wanted to tie back to the background, right? So we see that BHRF1 has been, its function has been, tied to prevent apoptosis within host cells, and that's been linked or characterized by other malignancies such as certain lymphomas, right? So specifically, this could extend a broader implication of developing like a strategic basis to combating the virulence and prevalence of the Epstein-Barr virus, which will in effect lower prevalence or combat malignancies and other lymphomas that arise out of those infections. Hopefully that kind of clears it up.
Excellent. Well, thank you for that. And now if there are any final questions, our 2 minutes is just about up, so please feel free. I'll have a few more seconds just in case anybody has some final thoughts. Otherwise, we will get started with the next one. All right, well, I think we will move on to the second video now. So our second video is the use machine learning to find significant features that discriminate between mild and severe rheumatoid arthritis by Amna Irfan under Dr. Das. So I'm going to get that loaded up. 1 second. Or, too confusing. All right. Well, without further ado, here is the second video by Amna.
Okay, hello everyone. My name is Amna Irfan, and I'm going to be talking about using machine learning to find significant features that discriminate between mild and severe rheumatoid arthritis. So just before I start, I want to give credit to my PI, who was Dr. Jishnu Das, and he was a University of Pittsburgh faculty as part of my summer internship program, TechBio REU, that I was just involved with in the summer 2020. So this title, our research project, sounds kind of scary at first sight, but what I want to do is kind of explain some of the context and background before we start talking about what we found in the data that we collected. So first thing I want to talk about is systems biology. And systems biology is just one field of biology where we look at the system as a whole versus the sum of the parts. And that is something that we talk about all the time. And just to give an example, I'm going to talk about this cake, right? So here, all the ingredients to make a cake is like butter, powdered sugar, salt, baking soda, baking powder, eggs, flour, milk, sugar, vanilla extract, and sour cream, right? And if I ask you to go study all of these different ingredients together, would you be able to understand a cake? Well, no, because a cake as a whole is different than just the sum of its parts, right? You need a specific order of the ingredients. You need a specific environment for those ingredients. You need specific times and ratios in order to make that cake. And so what systems biology is that we're going to study the cake itself versus just studying every little part that makes up a cake. So in our example, right, a system can be really anything in biology, right? A cell, an organism, the human body. But for us, the system is going to be an RA patient or a rheumatoid arthritis patient. And the sum of the parts, in contrast, is going to be every feature that leads to RA development, right? So this could be antibody responses, it could be prescription drugs, or it could be cholesterol levels, whatever that we think could be contributing to someone's different response in RA. So I just talked about that. So that already sounds like a lot, right? Having to understand all these different features at once to understand rheumatoid arthritis. And that's exactly what is the problem, right? That we cannot understand all those features at once. And that's what machine learning is for. It's a computational algorithm. So it's a subset of artificial intelligence that build computational models to predict or make decisions without being programmed to do so. So in normal computer science projects, what we do is we tell the computer a set of instructions and it executes that. It's basic. But in machine learning, what happens is we give the computer data and it makes its own instructions based on patterns or recognition sequences or whatever. So in our research project, we used Random Forest and SVM and other tools, but I'm just going to talk about these two main ones. So Random Forest is exactly what it sounds like, and it's just a forest. So a forest just means a bunch of trees, right? And that's what Random Forest is. It's a bunch of decision-based trees. And just to give an example, right here is a decision-based tree. So let's say I want to understand more about a specific individual. I can ask the question, is the gender of that individual male or female, right? And then I can answer yes, and I can go down, and I can answer no and go down. So I can choose one variable, or what we will call a feature, to be a node of the tree, and then keep going down with subsequent nodes, right? Age, SIBSP, right? And so on and so forth. So what we're going to do with Random Forest is we're going to give it some data, and it's going to build the decision-based trees on this trained data, right? Trained means it's using that data to make the model. And then the tested data is going to be used where we put new data into the model that was built on the trained data and see the accuracy of that data, of the tested data, to see the accuracy. So that's one model. And another machine learning model is called SVM. And SVM stands for Support Vector Machine. And so it also uses trained and tested data. But just to give some context of what SVM does, I want you to think about just regular graphs, right? In an algebra course, let's say you had x versus y. You had two variables, and you could plot those variables on a graph and see if it makes a line or a quadratic or a third exponent function, right? And so, for example, here, this population of bears over time, right? I have the year, and I can see how the population of bears is changing over time, and it just shows a little line. So what happens if I have three variables, right? Now I want to look at bears, time, and growth of the bears, right? I can do X, Y, and Z. But what about if I have more than three? I can't have more than three axes. We never talk about that. And so that's what Support Vector Machine is for. It's to help us make hyperplanes. And hyperplanes are going to be these planes that help discriminate our two major classes. And in our case, that'll be mild RA or severe RA, and make these support vectors, right? So the support vectors are going to go through those two major courses, and it'll try to find a margin between the most optimal hyperplane and the most outermost support vector. And once you do that, once you find the most optimal hyperplane and you find the support vectors, you make this model. And once you make that model based on the trained data, you can put, again, tested data into that model and see the accuracy of that tested data. So here are some of my data. So Lasso is just another machine learning model that I didn't get to talk about, but basically what it does is it just selects the most important features of a data set. So it could be like 5 or 6 features, or variables if you want to call it variables. And so once it does that, I put a random forest model on those features that were selected based on Lasso. And same thing here with SVM. And so AUC it just stands for area under the curve. And basically what AUC is trying to help us figure out is just the level of randomness, right? So in our permuted data set, what we did is we took our data set that we had and we just switched up our samples, right? We switched up the X column but kept the Y the same. And when you do that, it's basically random, right? Now it's not what we had measured. And so we expect our AUC value of the permuted data set to be around 50 or 60, right? It's about half the chance, which is just completely random, right? And that's exactly what we see with both data sets, is that it's around 50 or 60. But whenever we did our machine learning model, right, with Lasso Random Forest or Lasso SVM, we had seen a better AUC value, right? So that's like around 0.75, 0.75 here too. And that shows that the accuracy of this model is a little bit better than just random chance. Obviously, these are not the best results, but these are the results I had made over the summer. So some future directions is that the computational lab in Pittsburgh wants to continue finding other machine learning algorithms that will help increase that accuracy. And then once we can find the most accurate models to discriminate between mild and severe RA, we can use those models in conjunction with a physician's course of treatment so that RA patients feel a little bit better. Once we can find those important features of RA, we can target them, we can find treatments for them, and once we find treatments for them, people can start feeling better. And so the hope is that this is just some really far-fetched goal, but this is just the importance of highlighting why machine learning is so important, is that we can also use machine learning models to help us determine why some people who get COVID-19 feel really, really sick and get ill in the hospital, while other people who get COVID-19 don't feel anything at all. And so our hope was that once we understand more machine learning models, we can use those models to help understand significant features that lead to COVID-19 responses. And so that is my presentation today, and I hope you learned something new, and I hope you enjoyed it. So thank you so much.
All right. Well, thank you, Amna, for that presentation. And now we will have another 2-minute Q&A. And I think there were actually a few questions that were asked in the chat. Could you go into detail a bit about those a little bit more?
It was my question in the chat, so I'm just going to ask it again. Can you just give us an idea of what like the input and the outputs of your model are? Because I know that you gave us kind of like a very general look at it because you didn't want to get us lost in the weeds, but can you give me an idea of what goes into this model and what is it predicting, right? When you say area under the curve, what is it predicting and how are you comparing that?
That's a great question. So actually, the input of the model was a lot of features, like I had said, but just to give some examples, what was specifically happening was that each of the features were basically a metabolite that was already studied to contribute to rheumatoid arthritis development. And so those features included just like, for example, I don't know, I don't remember all the metabolites. There were a lot of different organic metabolites, but just like the ratio or the concentration of each metabolite that they thought would contribute to RA development. And so the output, once we use machine learning, create a model, whatever, the output of the data is just trying to see how well can we categorize those features to see if there's a difference in the ratios of those metabolites between mild RA patients and severe RA patients. I hope that answered the question.
Yeah, I think that was that's those are some important details that I appreciate you going into a little bit more detail on. That is so cool. Yeah, so I guess I just want to reiterate. I guess kind of the big picture of this is you're eventually going to be able to try to adapt this to predict whether somebody will or could develop rheumatoid arthritis. Is that correct?
So these are patients that already were diagnosed with rheumatoid arthritis. What we're trying to do is see, like, why do some people who get RA feel like not as bad versus people who feel really, really bad and need to take severe like painkillers? Like, what is it in their cellular makeup that makes those differences in symptoms and feeling of RA prognosis?
Right. All right. That's very interesting. I think that is super cool. All right, so our 2 minutes has ended now, and we will move on to our third video. Thank you, Amna, again for that. Let's see. Our third video is going to be Stress Promotes a More Aggressive Phenotype in Human Melanoma Cells by Abigail Fajardo under Dr. Villares. All right, and we will get started. Here it is.
Hello, my name is Abigail Fajardo, and I'm here to talk about the research I've had the amazing opportunity to be a part of. Under the mentorship of Dr. Gabriel Villares and in collaboration with the Ponce Health Sciences University in Puerto Rico. And we talk about how stress promotes a more aggressive phenotype in human melanoma cells. And so in a typical cancer situation, you have your cancer cells and you have your monocytes. And monocytes are immune cells that haven't differentiated or have gotten a role yet. These usually differentiate into macrophages, which later on attack the cancer cell and eliminate it. However, when your body undergoes stress, it releases certain stress hormones or catecholamines, for example, norepinephrine, which is what we're looking into for this experiment. And these specifically attack the monocytes. And once they reach the beta-3 adrenal receptors, these cause the monocytes to differentiate into tumor-associated macrophages. And these macrophages release cytokines, which are cell signaling molecules. But these cytokines have been known to promote tumor growth, angiogenesis, and metastasis. And once a tumor gets big enough, it is able to release cells into the bloodstream and later on lead to metastasis. Specifically in melanoma, metastasis would be either to the liver, to the lungs, or to the brain. And once a tumor has metastasized, the survival rate decreases to around only 20%. And the reason we're looking into melanoma cells is because our collaborators have done this experiment on ovarian cancer cells. And the cytokines they have been able to identify through their experiments, some of them have also been known to increase melanoma progression. And so we hypothesized that adding norepinephrine will release cytokines into tumor microenvironments that will promote a more aggressive phenotype in human melanoma cells. And we did this through a 24-well plate, in which we had melanoma cells, or the C8161 cell line, and macrophages, the U937 cell lines. And we had them in both a co-culture and monoculture scenario. The reason is because all cells release cytokines. We want to be able to identify cytokines that are only being produced when in conjunction of melanoma cells and macrophages together. That way we aren't just taking every cytokine produced into consideration. And for each group, we had one row doused with norepinephrine. That way we were able to tell the difference between when norepinephrine is present and when it's not present with cytokine production. And we took the supernatant from these well plates and put them in a cytokine array kit. And so what we were given is that we were given blank membranes. So it would be these squares just completely blank. And once we douse them with our supernatant, there are specific antibodies where each spot will attach to a specific cytokine. And that way we're able to quantify and identify the cytokines present. And as you can tell, when in the monoculture lanes, the amount of cytokines present is a lot less compared to the co-culture lanes. And if you look at the co-culture lanes specifically, in the red circles, you can tell that the cytokines present when norepinephrine is present are at a higher quantity, or they're present when they weren't present before. So they, due to our collaborators, they were able to quantify this data and identify cytokines present in a volcano plot. And what we're looking for are the cytokines being produced at a statistically significant level, which means over the p-value of 0.05, and with a positive fold change, which are these quadrants as shown here. And we're specifically looking for the cytokines only present in the co-culture lanes. And so what we found is that the main cytokines produced are listed here, but the ones of big importance are the angioprotein 2, which has been shown to help with angiogenesis as well as metastasis, and then IL-11, which has been shown to promote metastasis and tumor growth. And the reason this is so important is because a lot of cancer patients undergo a lot of stress through treatment. And stress has already been shown to negatively affect the body but now we are able to see that at a molecular level. And so while treating the cancer is a priority, we must also help treat the patient's well-being as a whole as well to give them the best chance for survival. And so the next steps we want to do is a new cell line in order to confirm that these effects occur in melanoma and not just one cell line, and then also do an ELISA assay, which will help us quantify and identify cytokines from each culture through using primary and secondary antibodies. And that is it for my presentation. Thank you.
Okay, Abigail, thank you for that presentation. And now we will open the floor to 2 minutes of questions. I can go ahead and get us started. So I know you did talk about it a little bit, but can you explain a little bit more how the volcano plot works? I think it's really interesting, but I'm not quite sure what I was how to interpret it, I guess. I can go back.
Yeah. So basically the volcano plot will plot the quantity or like I would say concentration of specific cytokines that were found once we ran the cytokine array kit. And so what we are looking for specifically, if you can like fast forward maybe like 5 seconds, there should be like a square around the top right quadrants. Yeah. And so we are looking for the cytokines that are shown as statistically significant levels. And we're also showing for a positive log fold change. So pretty much we're looking at everything in the top right quadrant. And those are the cytokines that have been shown that are present at significant levels that we should be looking towards.
Okay, very cool. That does make a bit more sense now. Thank you. Oh, and I think we have one question in the chat. I can go ahead and read it out. So Dr. Amin asks, what have you learned about the cytokines that cause this problem, and when are they released?
So all cells release cytokines since they're cell signaling molecules in order to communicate with other cells. But specifically these cytokines between the tumor-associated macrophages and the cancer cells are released once they are once the macrophages are in the presence of cancer cells. They normally don't release many cytokines before that. And so some of these cytokines have been known through either melanoma or other forms of cancer. They have been known to progress metastasis, tumor growth, and angiogenesis.
Okay, well, thank you for that. Our 2 minutes is now just about over, so we will move on to the next video now. But Abigail, thank you again for your presentation. That was very cool.
All right, thank you.
Okay, so our next video is the characterization of viral communities carried by male and female Aedes mosquitoes collected in Houston by Viet Tran, Martin Silguero, myself, Franklin Pham, Saley Davalos, Ana Hernandez, and Sarah Strobel, under Dr. Lares and Dr. Rosal. So 1 second, let me get this loaded up.
Hello, my name is Martin Salguero, and I will represent the University of St. Thomas in Houston, and I will be presenting our project, which is a characterization of viral communities carried by male and female Aedes mosquitoes collected in Houston. Our project focuses on characterizing the viral communities carried by different species of mosquitoes in Houston. While we know that mosquitoes carry important human viruses, like West Nile and dengue, the true collection of viruses that they carry is not well documented. Viral ecology in general is only now beginning to be characterized thanks to the sequencing techniques that sidestep the need for culturing. In our research, we are using metagenomic approaches to sequence the total genomic sample of mosquitoes and using a bioinformatics workflow developed in-house to identify viral sequences present in our samples. We are specifically looking at different species of mosquitoes commonly found in Houston, such as species from Aedes, Culex, and Anopheles genuses, and comparing the viral communities found in males and females of those same species. This work is relevant because Houston is an ideal breeding ground for mosquito populations due to a constant rain and accumulating pools of water the city experiences every year, and because they do carry serious diseases like previously mentioned, such as West Nile, which have been previously documented in our city. But we are interested in characterizing as many viral strains as possible in our samples and comparing the viral collections between sexes and across species. Our project does involve a little bit of field biology, such as capturing mosquitoes using traps, molecular benchwork, which includes nucleic acid isolation, and computational biology, which has to do with sequence data analysis, in order to reach our goal of characterizing the viral communities in these organisms. So moving into our methodology, our traps were set out at strategic points in the city, at first to coincide with areas where West Nile positive mosquitoes have been found, as seen in Figure 1, and then to other areas in the city to increase our data pool. Then these captured mosquitoes are brought into the lab where they are frozen until they can be processed at a later time. So once we have these mosquitoes in our lab, we separate our captured mosquitoes using morphological characteristics such as abdomen shape and coloration, striping patterns, and antenna and mouth parts. And we use that criteria to separate individuals and sort them by species and sex. So as we see in Figure 3, there is an example of how we differentiate between male and female mosquitoes, which is by their distinguishing antennae. So this antennae distinguishes between males and females by either being fuzzy or non-fuzzy, fuzzy for males and non-fuzzy for females. So as we can see in Figure 4, individual mosquitoes that have been separated by species and sex are then pooled into 10 milligram sets for nucleic acid extraction. We then grind these mosquitoes up to homogenize them and use protocol kits to isolate their total DNA and RNA. So part of the DNA that we extract, we use to verify the species by using PCR to amplify the cytochrome oxidase CoA1 gene. So once we have that, we send off our samples for sequencing to a private sequencing company and then use BLAST to confirm the genus and species of our samples. A sample of those sequences can be seen in Figure 6 and a gel electrophoresis that we create a lot in order to confirm the presence of those CoA1 gene sequences in that sample. On the other hand, RNA is set aside and used to make cDNA or complementary DNA to make a library of cDNA by RT-PCR or reverse transcriptase PCR. So we can sequence and transform the RNA into cDNA, which allows us to sequence and analyze the RNA viruses that may be present without having to work with an unstable and easily degradable sample of RNA. So once that's done, the total DNA and cDNA are pooled and sent off for sequencing using 454 technology. So far, we have completed one complete round of these experiments with male and female samples of Aedes mosquitoes, and when we received our sequences, our metagenomic sequence data needed to be sorted and curated in an automated way because of the sheer size of this data. This is why we devised a workflow in-house, as we see in Figure 5, that automatically converted 454 files to FAFSA files, and then automatically blasted each individual sequences in bulk. These results were then filtered for viral sequences hits, and all unknown sequences were separated into a new file for further analysis as well. So our initial results, as seen in Figure 7, show that we can sequence both DNA and RNA viruses. We were able to identify 51 viral families in our female sample and 40 viral families in our male sample. Interestingly, there is a marked difference in the abundance of viral types between the two sexes. We found a large representation of single-stranded RNA viruses in females compared to males, and males had way more DNA viruses. While differences between the sexes were expected given their different feeding habits, we need to do more sequencing experiments to determine if these differences are biologically relevant or a consequence of sequence biases in our experiments. As an initial proof of concept, our experiment shows that we can produce sequence-grade DNA and cDNA samples which we can use to identify both DNA and RNA viral sequences in our samples. We hope to continue to sequence our other samples and to determine if the viral distribution patterns are sex and species specific. These results will help us to better understand the true viral collections carried by mosquitoes in the Houston area.
Let's see, either Martine or Viet, I'm not sure which one of you is going to be responding to these, but regardless, Viet, all right. Yeah, so this covered, I think you said 80s, right? What are some other species of mosquitoes that you plan on working on in the future?
Yeah, so we've actually, so one of the main, the prevalence of the mosquito populations that we see in Houston, majority of the times we really pick out, I'd say around 80% of the species that we find tend to be Aedes aegypti. They're the most dominant strain that we have here, with a few occurrences in where we will catch Culex quinn and Anopheles, and there are other subspeciations, subgroupings for these other species. So it's really complex in terms of understanding what is our range in which we want to consider will be an independent group for us to start sequencing and classify. But definitely we're building up more samples for different species as we speak, and we were hoping to go run them very soon.
Very cool. All right, and I think there was another question in the comments, and I'll go ahead and read it out loud. So Dr. Amin asks, why would different sexes have different viruses? And why would males have higher DNA virus loads?
So what's super interesting is that different sexes will have different preferences in terms of their food and behavioral attributes and interactions within their environment. So when you think about females and males, you start to see that they have different behaviors. They choose to do different things in terms of their resource gathering. Females have a priority to create eggs, and therefore it's tried and tested, and it's shown that it's actually the females that will bite and inject themselves and expose themselves more as a vector for viruses. As like they suck up blood from other animals, and they use the iron within that blood to create eggs. And what you'll see is there's a great diversity of viruses within females. Now, in regards to Dr. Amin's question, why would males have higher DNA virus loads? I don't know. That's a good question. That is a very good question. I can say that DNA viruses tend to be more stable in terms of their structure in comparison to RNA-based viruses, but that's not so the case in terms of when you're looking at the female population. You can see that we have a lot of RNA-based viruses. But I don't know, it's a very good question. That's something that we have to explore more.
I'll chime in. This is Dr. Larios here. Just to remind everybody that it is important to keep in mind that this is based on one single sequencing experiment, and some of the discrepancies that we see between the two groups might be due to sequence bias, et cetera, right? So we do need to definitely replicate this in more groups to see if it's a true effect and a feature of the sex difference, or if it's just an artifact of the sampling.
Okay, well, Viet and Dr. Larios, thank you for that. And now we will move on to our second to last video. So now it is stress promotes a more aggressive phenotype in human melanoma cells, the future experiment by Leila Barkude under Dr. Villares. 1 second, let me get it loaded up. Okay, and here we are.
Leila Barkude. I'm a part of Dr. Villares' research group regarding melanoma cancer, and today I'll be telling you how stress promotes a more aggressive phenotype in human melanoma cells. Specifically, I'll be focused on the future experiment. So if you haven't already, please go back and watch my lab team member, Abigail Fajardo's video. If you have not watched her video, I would urge you to do so because this video would make more sense. And so, as I said before, I'm talking about further research. And so one of the things that we're trying to do is try to use different cell lines. And currently we're trying to do our data with the cell line A375, and we'll report the new findings once we have them. And as Abigail said, the sympathetic nervous system releases two hormones, one of which is norepinephrine and the other one is epinephrine. We're currently using the norepinephrine in our experiments. However, we're trying to use epinephrine in future experiments. And I hope that in Abigail's video, you saw that we are using cytokine array kits, which basically have different antibodies for different cytokines on them. And through these antibodies and secondary antibodies, we can see dots. And these dots represent the presence of cytokines. And to make sure this data is correct and to verify it and to further our research, we're trying to use an ELISA assay. An ELISA assay is a multi-titer plate that is filled with solutions and serums and can be easily prepared and tested. To take a closer look into the ELISA assay, we're going to take a closer look into each well. Now, each well has a specific antigen or a cytokine in our case, and which is done by a manufacturer. Now, we inject into the well a specific serum that has antibodies, and the specific antibodies will bind to the antigen. However, other antibodies will get washed away, as you can see in the third well. Next, there will be an injection of secondary antibodies that have enzymes on them. And these antibodies will help us to detect and see the cytokine's presence. After our secondary antibodies are injected, and washed away, the unbound ones washed away, there is an enzyme or color substrate that is put in that interacts with the enzyme on the secondary antibody, and that produces color. And you can see here in our ELISA well plate, there is a shades of color indicating the presence of the specific cytokines that we are looking for. And if they do have a color, then our specific cytokines are present. And so the cytokine array kit will be proven. Thank you so much for listening to this video. I hope you found it helpful, and I hope that you found the collection of our videos for Dr. Villarreal very helpful. Thank you.
Okay, Layla, thank you for that presentation. And now, again, we will have a 2-minute Q&A.
I have a question. Dr. Roselle? Layla, are you around? My question is, can you use the ELISA assay to do any quantification of how much cytokine you have?
I can help answer that. So the ELISA assay will help us both identify and quantify the amount of cytokines that will be present in our experiment.
Okay, how do you do that quantification?
So once we run the ELISA assay, we will most likely, like we did with the cytokine array kit, we will send our information to our collaborators in Puerto Rico, and they are able to quantify all the information, just send us everything back.
Okay, so it's not just a colorimetric change?
The colorimetric change, like, I'm not sure if you were there for my presentation, but if you saw the cytokine array kit, there's different shades of black, kind of giving you a gauge of the quantity of cytokines present. And so it'll be something similar here, but to quantify it at a numerical level, we need to send it to our collaborators.
Okay, because I thought with an ELISA, it has the enzyme actually produces change in color, and you can use the spectrophotometer to actually quantify that change in color. Okay, all right. Thank you.
All right. Yes, thank you, Dr. Roselle, for those questions. If there are any further questions, we have a few more seconds, so please do ask. I will give a few more seconds for you to do that. Okay, if there aren't any further questions, then we will move on to our last video, actually. So our final video for the day in this session will be toluene exposure and the resulting toxicity effects on fly fecundity and offspring development in Drosophila melanogaster by Steve Trong, Francisco Lopez, Carolyn Lawrence, Imad Ullah, Michelle Ballinger, Need Thomas, Thomas Ferguson, and Tyler Serrano.
Hello, my name is Steve. My research is on the toluene exposure and resulting toxicity effect on fly fecundity and offspring development in the Drosophila melanogaster. Houston, Texas is a leader in the oil and gas industry, and they often face health hazards due to byproducts and pollutants by oil and gas refineries. Several years ago, an unauthorized release of chemicals by the company ITC polluted the air within the greater Houston area. The same company was recently responsible for an enormous explosion that exposed the greater Houston area to chemicals such as toluene. Toluene is a volatile hydrocarbon that is found in household products in small, unharmful concentration, and is classified as non-cancerous but air toxic. Chronic toluene exposure has been established to affect several organ systems as well as fetal development. Our purpose is to investigate the effects of toluene on the model organism, the Drosophila melanogaster. Since D. melanogasters have similar gene homologs to humans as well as nervous system structure and function, we infer that the exposure to toluene will affect the reproductive system in the fruit flies. This study hypothesizes that the exposure of toluene will negatively affect the reproductive and nervous system in the fruit flies. To observe these effects, a continuous flow system that delivers known concentrations of toluene to the populations of the D. melanogaster was developed, and in Figure 1 are the schematics of the system that is used to expose the fly to the chemical. We interpolated the lethal exposure of toluene that kills 50% of the populations between 1,000 and 1,200 parts per million. Here in Figure 2, we can see the relationship between mortality and concentration. As toluene concentration increases, so does mortality in the female sample. The resulting offsprings are counted at egg, larval, and pupal stages. Our results show that as toluene concentration increases, there is a decrease in fly fecundity and offspring survivability, indicating the effects of toluene are dosage dependent and have detrimental effects on fly development. The results in Figure 3 show that as toluene concentration increases, the average number of eggs produced by the female decrease. In Figure 4, the exposed females on average produce fewer eggs per day than the control flies. The exposed males have a peak of fertility on day 3. In Figure 5, there are two takeaways from this graph. The larvae of the exposed female eclose earlier than the control, and the larvae of the exposed male eclose later than the control flies, represented by the early and late peaks respectively. As we expose flies to toluene, we can begin to understand the effects of toluene on survivability and reproduction. We establish a dosage-dependent relationship. As concentration of toluene increases, the fecundity of the adult flies decrease. Because humans share gene homologs and by default DNA, we would expect to see decrease in fertility in humans as well. We predict that the exposed males have a different LE50 than the females because the eggs and larvae which eclose were significantly higher than that of the females, possibly indicating that female reproduction is more sensitive to toluene exposure. We would expect that if this pattern were to continue, there would be more pupae and adults eclosing than the exposed females at this concentration. In conclusion, we have been able to observe that toluene decreased fly fecundity, its effects increasing directly proportional as concentration increases. And we would expect to observe the same in human fertility.
All right. Thank you, Steve, for that presentation. And now we will move on to the 2-minute Q&A. So I will go ahead and get us started if that's okay. So actually, how did your group decide on the concentrations of toluene to administer to the fruit flies? And if possible, what were these concentrations like analogous to the concentrations that were in the air after that explosion? Are there any members of this research group present?
Looks like I'm the only one.
Okay.
Those are good questions, Trey. Our concentrations right now, I think, are a little higher than what the concentrations that were released were higher than what we are testing. However, they dissipate rapidly in the air. Okay, when they're released through a big explosion or something like that. What we're delivering to the flies is a little bit high, but we're trying to find a concentration at which they don't all die, but it's like a sublethal concentration. That's why we look at that lethal concentration to where 50% of the population die, and then we do our test below that, so that not everybody is dying, but you are seeing effects. And what Steve was talking about there is that we've determined with the females that's in our system about 1,200 parts per million, which is high, but it's not killing the fruit flies. At that, not killing all of them at that concentration. So, but we are seeing effects on fecundity at that concentration. And the thing about males is that we've just started testing the males, and so we're exposing males and then mating them to unexposed females. So I think that we're seeing a higher effect on the exposed females than we are to females that have been mated with males that have been exposed. So we're trying to look at all kinds of combinations. And I think that the reason that, I can't remember your question. I was reading Dr. Amin's question about why females would be more sensitive. I think that females are more sensitive because they're at that point of exposure, they're making eggs. And most of the physiology is turned toward making eggs at that point. And so that's why I think the reproductive system is so affected. And then Trey, what was your question? I can't remember.
Oh, let's see. I did ask about whether the concentrations were analogous to like the concentrations released from that chemical explosion that was mentioned in the intro.
Yeah, those concentrations were really high, but like I said, because they were released into the environment, they dissipate very, very quickly. Okay, so you'd have to be very close by to get a very large exposure. Daily workers that work around those kinds of industries are getting somewhere around 200 parts per million daily. And that they are seeing effects, but it's a cumulative effect in those workers.
Very cool. All right. Anybody have any additional questions?
For Steve?
Yeah, I'll give a few seconds just if we have anybody else who has anything additional to add. All right. Well, if not, then we will move on. I believe that was our last video for the day, actually. So one moment. I will just say some final things, and then we will end this session. So I would like to thank all the presenters for submitting videos. And if you're able to check out any of the other sessions, they will be recorded as well, in case you weren't able to view them live. And also we have a post attendance form that I will send the link to in the chat once more. And please fill that out for feedback, both for ASBMB and the presenters. We would greatly appreciate it. And thank you so much for attending. This has been very fun, and I know this is a little bit different to have a virtual bears compared to an in-person bears, which I'm sure many of you upperclassmen remember fondly. But those freshmen out there, you will hopefully get to see an in-person bears next year, or whenever possible. But in the meantime, this has been very enjoyable. So thank you all again for attending, and that is it. Thanks for coming.
Well done, Trey. You guys did a great job. Thank you.
Appreciate it, Trey.
Thanks everybody. Have a good one.